Hi, my name is Daniel, and today I would like to talk to you about the attractive and repulsive cues used for axon guidance versus cell migration. Neuronal migration and axon guidance share many features, for example, that they rely on external guidance cues, and some authors even regard axon growth as a specialized form of cell migration. However, there are also important differences in the cell biology of these two processes, which I want to point out today using examples from different parts of the developing nervous system. We will first talk about some of the guidance cues we encountered throughout this class, and then compare the cell morphology changes these molecules cause. At the end, we will try to understand how intrinsic signaling cascades connect the extrinsic cues and the morphological changes. We talked about neuronal migration mostly in the context of cortical development. There were two types of migration: radial migration of excitatory glutamatergic neurons, which are born in the ventricular zone of the dorsal telencephalon and then migrate along radial glia cells to form the layers of the neocortex. Cortical interneurons, however, develop in the ganglionic eminences in the ventral telencephalon and first have to migrate over long distances before integrating into the cortical circuitry, which is called tangential migration. In both cases, combinations of attractive and repulsive cues play an important role. While the attractive reelin signal is critical for migration of the excitatory neurons, the driving forces for the long-range interneuron migration are repulsive afferent signals in the ganglionic eminences and the attractive CXCL12 cue expressed by the marginal and intermediate zone of the cortex. We also discussed the repulsive semaphorin signal, which prevents migrating cortical interneurons from invading the developing striatum. However, striatal interneurons do not express the semaphorin receptor, making them non-responsive to this repulsive cue, which already hints at how important intrinsic signaling and receptor patterns are for these guidance mechanisms. When we talked about axon guidance, we discussed the midline crossing example of commissural axons in the spinal cord. Before midline crossing, these axons are attracted by ventrally secreted molecules like natrin. But when the axons reach the midline, there is a dynamic shift in receptor expression, now making the axons responsive to repulsive cues, which in this case are slit and semaphorin. One thing we notice when comparing these examples is that there are so-called canonical guidance cues which influence both neuronal migration and axon guidance and classically comprise semaphorin, natrin, slit, and afferent. In fact, it has even been shown that their effects go far beyond guidance and the nervous system. To better understand how the same canonical guidance cues can govern both of these processes, it is helpful to take a closer look at how neuronal migration and axon guidance are different on a cell morphological level. While both neuronal migration and axon guidance depend on a stabilizing microtubule network and a more flexible actin network in the exploring filopodia, one key difference is that the growth cones in axon guidance act as independent units. In neuronal migration, on the other side, microtubule and actin networks are connected to the nucleus by a so-called microtubule cage. In axon guidance, detection of external cues leads to filopodia extension and elongation of the axonal microtubule network. In cell migration, on the other side, elongation of the microtubule network is followed by a process called nucleokinesis. During this essential step, the nucleus translocates into the leading process, followed by the retraction of the trailing process, so that the neuron ultimately migrates. So now the remaining question is, how can the same canonical extrinsic guidance cues lead to these different types of cytoskeletal rearrangement? But before we move on, I want to quickly show you that the morphological reality is much more complex than what we just discussed. The morphology of the leading process varies between different types of neurons. It has been shown that tangentially migrating cortical interneurons generate a branched leading process as part of their migratory cycle. The two branches show exploratory behavior and seem to allow the migrating cells to rapidly change their direction, which is particularly useful for the radial invasion of the cortical plate. So, what is the missing link between these complex morphological changes and our earlier discussed extrinsic guidance cues? The answer lies somewhere in the intrinsic signaling pathways and receptor patterns of these cells. When looking at the molecular signaling cascades in axon guidance, we can see that the signals from our four canonical guidance cues, ephrin, natrin, SLIT, and semaphorin, get integrated at the level of GTPase activity and GTP levels, which then leads to cytoskeletal rearrangement. When comparing this to the signaling in neuronal migration, we can again see our canonical guidance cues, and we can also find many similar downstream molecules like CDC42, RAC, and Rho. While we have learned that different receptors, for example the semaphorin receptors neuropilin and plexin, can be used to activate different downstream effector pathways, the exact signaling cascades mediating the cytoskeletal differences in neuronal migration and axon guidance are not entirely understood. Molecules like CDK5, which plays a critical role in migration and has been extensively studied in this context, has also been shown to play an important role in axon patterning and many other processes beyond that. This brings us back to this image from the beginning, showing that the function of many of these guidance molecules and their effectors probably goes beyond just guidance. Thank you very much.